that the name of my university is a little bit weird, and ABC stands for the name of the cities where the university has campus. So A for Santo André, B for São Bernardo, and we don't have a campus on C, but anyway, that's the reason for ABC. Okay, I'm really glad you're all here to watch my presentation and see me talking a little bit about the costs of mutualistic interactions. So just when you think about mutualisms, you usually think about cute things like this clownfish and anemones, and pollination interaction, interaction between ants and honeydew insects. So mutualisms are really common interactions, and they are widespread. They happens everywhere in the globe. They happens in water and land, and they are also hypothesized to be a driver of diversification. So here we have two studies, two articles mentioning how this mutualisms enhance plant diversification or primate diversification, but there's a lot of others examples showing this pattern. But also mutualisms happens in plants. So I'm going to focus here on plant interaction, and plants interact with a bunch of mutualisms at the same time. For instance, they can have interactions with rhizobia or mycorrhiza on the roots, and they exchange nitrogen and they provide carbon for these mutualistic partners. They can interact with ants. So ants confer defense to plants against herbivores. And in turn, they provide carbon or nectar for ants. And they also interact with pollinators, angiosperms interacting with pollinators. So they provide also nutritional rewards, nectar, pollen, oil, and they get the transfer of the gametes. And there's a cost. We usually think about the direct cost. So, for instance, in pollination or in plant defense, bees need to find plants, ants need to find plants, and plants also pay a cost in these mutualistic interactions. They are providing nutritional rewards for all of them. But there's also one cost that I less study yet, which are the indirect cost of mutualistic interactions or the indirect effect of mutualisms on another mutualisms. What is this? So, for instance, this is the effect of one mutualistic partner on the other mutualistic partner, and this could have an effect for plants. So here we have the indirect effect of rhizobia on pollinators, and there are some studies showing that plants with rhizobium have a larger floral display, so they have more flowers, which attract more pollinators. So this is not a cost, this is an indirect benefit for pollinators. But they can also have indirect effects or indirect costs in this case, direct cost on rhizobia because they're providing the same resource and plants are providing the same nutritional resource for ants and for pollinate for ants and rhizobia. So there's a study showing that plants with rhizobia provide less nectar to ants, which could decrease the defense. And there is also another indirect cost, indirect effect of ants on pollinators. This is the cost that I am talking to you today, and this is the cost I'm most interested. And there's also the indirect effect, of course, of ants on pollinators, but there's a direct effect of ants on pollinators because ants can repel pollinators. So I'm going to talk a little bit about this interaction mainly. So when you think about pollination, so pollinators visit the flower, they get the pollen, and then transfer the pollen to another flower, which could generate a seed set. And this is a good interaction for pollinators and for plants because plants get the pollen transfer, pollinators get the nutritional resource, usually pollen or nectar. From the protective mutualisms, I'm going to focus here on the protective based on extrafloral nectar secretion because other kind of plants they can house ants in structures called domatia. This is not what I'm going to talk about here today, so I'm going to talk only about plants offering extrafloral nectar secretion. So in this case, plants have these extrafloral nectaries or EFNs. They can vary in structure, in size, in shape, and concentration of nectar to secrete. Here we have a droplet of extrafloral nectar in a chemicrystal nebulous. And this nectar attracts ants. Ants patrol the plants. While patrol the plants, they can find herbivores, and they usually repel these herbivores or they consume these herbivores. And here we have a video of a lab mate of mine showing this effect of ants repelling a herbivore. And this is a really cool video. This happened in the Amazon forest. So they all have benefits. So ants get the benefits of nutritional resources, and they provide protection against herbivory. So what about indirect costs? So here we have a video of a bee, Algoflora bee species. She is actually getting floral resources, probably nectar, and we can see an ant repelling this bee. So there's an ant here repelling her. So this is an indirect effect because ants can repel these pollinators. And the first chapter of my thesis, I conduct a meta-analysis to study the effect of ants on pollination in general. This study is under review right now. And in this study, I got 134 effect sizes from 28 studies and 25 plant species. I'm going to show here just a subset, just one analysis of this meta-analysis, which is the effect of ants on different floral visitors. We found the effects of ants on bee visitors and butterfly species. And this is the effect sizes, HGG, values lower than 0 show that ants have a negative effect on pollinators, on bee or butterflies. For instance, they stayed less in the flower, so short visit. Positive effects, ants have a positive effect on bees or on both butterflies, so they stayed more in the flowers with bees or butterflies. So let's see what I found. So I basically found that ants have a negative effect on bees. So bees usually stay less in the flower, for instance, and they have no effect on butterflies. So this conflict between ants and bees, we saw low impact on plant performance, actually. And this could have been because of the evolution of strategies reducing this conflict. And one strategy that could reduce these conflicts, for instance, the rise of plant protection mutualisms on plants not pollinated by bees. And which plants are pollinated by bees? So we are working with bus-pollinated plants. These plants have a trait called poricidal anthers. So these anthers are like a bottle of water, for instance, and the pollen are inside this anther, and they're only released when bees go there and vibrate the anthers. And besides bees, only 2 species of Diptera can vibrate the anthers. So basically, just bees can vibrate the anthers and release the pollen. So these plants are mandatory dependent on bee pollination. So we have here non-parasital flowers, and non-parasital flowers can be pollinated by hummingbirds, bees, butterflies, and these parasital flowers, so which are the plants that have these parasital anthers. And if ants bothers bees, there might be a high conflict or a high effect on these parasital flowers with a high cost for plants because these plants requires bee pollination. And most studies show this from ecological scale. And what we try to do is to show how this conflict happened on evolutionary scale. So our hypothesis was that this ant-bee conflict might constrain the simultaneous occurrence of traits mediating protective mutualism, so extrafloral nectaries, and bus pollination in angiosperms. And for that, I got the all angiosperms that occurred extent angiosperms. So we got the plant names from the queue, Plants of the World online. And the data set of plants with extraphloral nectaries we got from Dr. Marjorie Weber. And she has this really cool website with a huge data set of plants interacting with ants, so plants with extraphloral nectaries. And I consider this, if at least one species of the genus have extraphloral nectaries, I consider that the whole genus could have extraphloral nectaries. And for buzz-pollinated plants, I got this data set from Dr. Avery Russell. And this study is on this preprint server. You can all have access to this buzz-pollination data set. And again, I consider that if at least one species have pursidoanthers, the whole genus could have pursidoanthers. And I use this phylogenetic tree by Steven Jensen to plot this data. So what did I found so far? I found that so we have plants, the first part, plants that are not buzz-pollinated, plants that do not have extraphloral nectaries, plants that are not buzz-pollinated but with extraphloral nectaries, so plants interacting with ants, buzz-pollinated plants but without extraphloral nectaries, and buzz-pollinated plants with extraphloral nectaries. And in the y-axis, we have the number of plant genera. So basically what I found is that most plants are not involved in protection mutualisms by ants or buzz-pollination mutualisms. Only 6% of plant genera are involved in protection mutualisms with ants, but not buzz-pollination. Most of them are Fabaceae, 15% of data, Euphorbiaceae, and Bignoniaceae. Regarding buzz-pollination, we have that 5% of all plant genera are buzz-pollinated, and most genus are Melostomataceae, Araceae, and Ericaceae. And only 45 genera, or 0.003% of all plant genera are involved in ant protection with extraphloral nectaries and buzz-pollination. Most of them are Dipterocarpaceae with 8 genera, Fabaceae 5 genera, and Polygalaceae 3 genera. And these are the phylogenetic tree with this data. I know it's kind of a lot. There is 14,000 genera plotted here. So in blue, we have the extraphloral nectaries. In light blue, the absence of extraphloral nectaries. In darker blue, the presence of extraphloral nectaries. So you can see that they are all widespread. They have independent evolutions on the angiosperm phylogenetic tree. And in yellow, orange-ish, we have the pursidoanthers. In light orange or yellow, we have the absence of pursidoanthers, so plants that don't require this buzz-pollination. And in orange, we have buzz-pollinated plants. So again, this is all widespread on the phylogenetic tree and with multiples independent evolutions. So we wanted to test if there's a correlated evolution between extraphloral nectaries and pursidoanthers. So we test an independent model in which these traits evolved separately. So here we have non-pursidoanthers from pursidoanthers and non-extraphloral nectaries to extraphloral nectaries. And but the model that was has the lower AIC was this one. So this was the favored model, the dependent model, showing that there is a correlated evolution between extraphloral nectaries and pursidoanthers. So we have here non-pursidal plants without extraphloral nectaries and non-pursidoanthers with extraphloral nectaries. So we have here the evolution of extraphloral nectaries. And we show that there's a higher chance of plants losing the extraphloral nectary, but there's a chance of plants gaining this trait. Regarding pursidoanthers, we have here the evolution of parasitic enters in plants without extraphonectaries. So there's a low chance of plants gaining parasitic enters, but there's still a chance, but there's a higher chance of plants losing to rate. And this is a really cool result. This is the evolution of parasitic anthers and exophorenectaries. And it seems that there's a high chance of plants losing this trait and becoming like non-parasital. And there is no chance of plants gaining the parasital trait from plants that already have exophorenectaries. So we are showing here that exophorenectaries constrain the evolution of parasital anthers. And here we have this parasital plants and without exophorenectaries to parasital plants with exophorenectaries. So there's a chance of plants gaining this exophorenectaries, but there's a higher chance of plants losing this trait. So probably the evolution of plants with exophorenectaries and parasital anthers, so both pollination and protection by ants, happens from this way because this parasital anther does not constrain the evolution of exophorenectaries, which is a really cool result. So we found evidence for the hypothesis that the conflict between ant and bees constrain the simultaneous occurrence of this trait. And this happens because the ecological costs, especially the ones that compromise fitness, in this case plant fitness, might be an important selective force. And the costs that we measure today, they might be low because they might have been minimized over evolutionary time. But one thing that got me thinking was, okay, so there's a conflict, this conflict is intense, but why some plants, this 4 or 5 genera, they have both exophorenectaries, they are protected by ants, and they are pollinated by bees. So what happens in these 45 cases? So we had a few hypotheses about this. Maybe these plants are a little bit less dependent on the pollination. For instance, they might be self-compatible. So they can have some fitness even in the absence of a pollinator. Or bus pollination might be a really efficient system, and even if ants are there, bees can have and even short visit by bees, they can be enough to generate some seed set and fruit set, some fitness. So they might be a really efficient system. But they also might have been involved in areas with a high herbivory pressure, but also a high abundance of bee pollinators. So okay, so what's next? What's the other questions we had from all of this results? Basically, we were thinking about how this exophorenectaries change flower phenotype because this everything that I showed here, the results I showed here, are on a macroecological scale. We worked with all the angiosperms, but what if we think just in one genus? What happened there? So how this exophorenectaries can change the flower phenotype in this genus? So we chose Chamaecrista. And other thing was that how this herbivory gradient can affect the evolution of exophorenectaries and flower traits, considering that in places with a high herbivory pressure, plants might invest more in protection, so they might have more exophorenectaries, and this could impose a higher cost on the pollination. So how does this happen? And this is these two questions I'm answering with the help of the Rosemary Grant by the Society for the Study of Evolution. So thank you, SSE. So to answer these questions, I went to the Royal Botanical Kew Gardens, where I stayed for 2 months. So I just came back from there in September. And Waikil Garden, so basically they have a huge amount of plants there. It's the second largest herbarium on Earth. They have 7 million preserved vascular plants. And why Chamaecrista? So Chamaecrista is a genus with 330 species. They are widely distributed, mainly in America. And this is the key, the Chemicrista distribution uh, in Kew Gardens. So Kew Gardens, they have almost 4,000 specimens, and each dot here represent a specimen available at Kew. And they also represent the distribution of Chemicrista across the globe. So we don't have Chemicrista in Europe, we don't have Chemicrista in Asia, but we have Chemicrista in Americas, Africa, and Australia. All species are both pollinated, so they all could have this conflict between bees and ants. There is a diversity of flowers. Okay, you can look this and look how they are all yellowish. They are not, but they are. They have some differences, and there is no scale on these pictures, but they vary in size. They vary a little bit in color, so they vary from yellow to reddish, orange. They vary in size and things like that. I'm going to show more traits in a few slides. And approximately 70 species have extraphilonectaries, and these extraphilonectaries, they also have a high diversity, uh, both in location. So some extraphilonectaries are located only on vegetative tissue, close to leaves, but some Chemicrista species have extraphilonectaries close to inflorescences, which could be a higher cost to pollination because ants would be in a close contact with pollinators, right? Um, so this is me working. So I measure some floor traits. So we have Chemicrista depauperata. So just to show that there is a variation in plant species, so there's, we can see how small these species are. And okay, so from all 300 species, I collect, there is 231 species in the phylogeny. So this is the current phylogeny of Chemicrista with 231 species. In black tips, so I know you cannot read that, I cannot read that, and this is not the most important thing, and I'm just here. And this figure is just to show the species that I have data and things that I don't have data. So I have data for all these species in black, and I do not have data for the species in red. So I need more data for this code. But so far, so in the phylogeny, there are 231 species in the phylogeny. And I have data, I sampled 156 species, which corresponds to 68% of our phylogeny. And of course, so there is 75 species that I did not sample. And from these 156 species, I sampled 105 species at Kew Botanical Gardens. So I sampled a lot of species there in this 2 months I stayed there. But there's a potential to measure a little bit more. From the 75 species that I don't have data, actually there's 38 species in São Paulo herbariums. And me and my lab mates, we are going to these herbariums in São Paulo to measure Chemicrista traits, floral traits, and extrafloral nectar traits. So with this, if I can add this 38 species, I would have 194 species from this 231 species in the current phylogeny, which could correspond to 84 species. I might not have data for all of them, but up to another 194 species. So, okay, so I showed you where I've been and the amount of data that I have sampled. But I'm going to focus on this last part of the talk on trying to answer these questions. Unfortunately, I don't have real data yet for this. I have the hypothesis. I'm going to show you my predictions because we are still collecting the data. So probably in the near future, I'll have more to show you. But until I finish all the data collection, I thought it was easier to show you my predictions because we can all work together. If you have any feedback and suggestions, I'm glad to hear it from you. So, okay, so the first question was how does the exophaneacteries change flower phenotype in Camecrista? So for that, we have two hypotheses. The first one is that plants with exophaneacteries should have traits that produce and access to flowers compared, of course, to plants without exophaneacteries. And also that plants with exophaneacteries should have traits that reduce pollen limitation caused by ants, especially if ants can access flowers, right? So I'm gonna show you predictions for each of these hypotheses. So the first prediction is related to trichomes. So trichomes might reduce and access to flowers. So here we have Camecrista aurivula. They have a huge amount of trichomes. You can see the petal of the flower. And trichomes might prevent ant access. They might be hard for ants to walk and reach flowers with this huge amount of trichomes. So I'm gonna show you a bunch of figures right now with my predictions. They are all in figures. And from now on, we will always have exophore nectaries, plants with exophore nectaries, plants without exophore nectaries on the x-axis, and the trait I'm talking about on the y-axis. So basically, we think that plants with exophore nectaries might have more trichomes, or they might have trichomes when compared to plants without exophore nectaries, because trichomes might be um, hard for ants to walk, might make ants' life harder. Um, Also, we were thinking about the pet cell size. So the pet cell is the size between the flower and the branch. Um, so maybe because there's like a spatial separation between flowers and branches. So plants with exophore nectaries, they might have a higher pet cell than plants without exophore nectaries. And also, finally, for this hypothesis, the type of inflorescence. So Camecrista can have isolated and flowers or inflorescence. In this case, the left case, we have plants with a small display. So just one or a few flowers open at the same time. On the right, we have a large display. So this might be more attractive to pollinators than the Camecrista dentacta with a small display. So it's possible that plants with exophore nectaries, they might have a larger display or more flowers because these plants might be really attractive to pollinators. So they have a lot of pollinators going there simultaneously. So ants might have a lower effect on this case when plants have a smaller, no, a larger number of flowers. So these are my three predictions for this hypothesis. For the second hypothesis, we have these plants with extrovert nectaries. They have traits that reduce pollen limitation. So the predictions are related to flower size. There's some studies showing that larger flowers, they are less pollen limited than smaller flowers. And because I think it's a little bit hard to see the size of the Camecrista nigricans flower on your left, I'm gonna zoom in. And this is the flower, so it's less than this is 1 millimeter. So this flower is less than 1 millimeter. So it's possible that plants with exophore nectaries, they have a larger size than plants without exophore nectaries. The second prediction is related to the stigma diameter because plants with a higher stigma diameter they can receive more pollen than plants with a less stigma diameter. So this is the measure. I didn't measure this in field work. I measured using herbarium data, but I didn't take any pictures of this measurement. But basically, what we think based on this larger stigmas receiving more pollen grains is that extra plants with extra nectaries they might have a larger stigma size when compared to plants without extra floral nectaries. We also measure the pore area of the anthers. So here we have an anther and a porcidal anther, and we measured this area because it's possible that plants with a larger pore area, the pollen might be released easily when they have a larger area compared when they have a small area. So we think that plants with exophanectaries might have a larger pore area, so pollen could be released easily or more easier when compared to plants without exophanectaries. We also think about we were trying to think about this self-compatibility and maybe plants with extra nectaries they might have be less dependent on plant pollination on bee pollination. They might be self-compatible, for instance. And of course, we don't have a way to measure this in herbarium data. So we were thinking about a proxy we could use to estimate this compatibility. And we were thinking about the gynoecium, a ratio between the size of the gynoecium and the larger anther. So in this case, we have Chamaecrista nigricans. You can see that the stigma is in here. So it's basically the same size or a little bit smaller than the anthers. So they might receive self-pollen. They might be able to self-pollinate it. This is a hypothesis. But there's also other plants that have a larger gynoecium compared to the anther. So in this case, this gynoecium or this stigma probably will not receive self-pollen. So this is the proxy we were thinking about compatibility. So in this case, we are thinking about calculating this ratio. So if this ratio is lower than 1, we might have plants that are self-compatible. And if this ratio is larger than 1, we might have plants that are not self-compatible. So in this case, plants with exophanectaries will have and this smaller ratio. So the gynoecium will be close in size to the anthers when compared to plants without exophanectaries. So these are all the predictions for this hypothesis that plants with exophanectaries should have traits that reduced pollen limitation caused by. Regarding the second, so I thought I talked a little bit about the first question. Regarding the second question about how the herbivory gradient could affect EFN and flower traits in Chamaecrista, we are so I just want to show you here that this is a hotly debated theory or hypothesis about herbivory increasing towards lower latitudes. So this study here, published in 2015, they showed exactly this. There's a higher herbivory rate in plants close to the equator line, so in lower latitudes, but this is a hotly debated theory. And we also had the same hypothesis for the other question, but we think that all these effects they should be stronger towards lower latitudes. I won't show you again all this justifications or these figures one by one. I just want to summarize here. So we will have in all figures the latitude in the x-axis from 0 to 90. And on the y-axis, again, the same, the traits we are interested. And we'll have two lines, different in color. So orange represents plants with extrafloronectaries, and in green, plants without extrafloronectaries. So we predict that for trichomes that there might not be an effect with trichome plants without extrafloronectaries, but this effect should be stronger for plants with extrafloronectaries, especially in lower latitudes. So plants in lower latitudes, they might have more trichomes than plants in higher latitudes. The same for pet cell size. So plants might have larger pet cell or than plants in higher latitudes. And the same for display. The plants should have more flowers in lower latitudes than in larger latitudes. What I didn't told you was that we are collecting all this data. We are also collecting the place where all these plants were collected. So this is how we are using the latitude. Some specimens, of course, they provided the latitude, some authors, but others, they just provided the place. Oh, this plant were collected in this area or this park. So we are getting the latitude from these places too. And for the second hypothesis, again, we have the latitude. I forgot to add the x-axis, but we have, again, the latitude, lower latitudes, higher latitudes. And again, in orange, plants with extrafloronectaries, and in green, plants without extrafloronectaries. So regarding floral size, so plants might have a larger flower close in lower latitudes compared to smaller latitudes. A sigma should be larger also in lower latitudes than in higher latitudes. The pore area should also be higher or larger actually in lower latitudes and higher latitudes. And we don't know about this latitude in this trait, but maybe this they might have similar size. So they might have a similar size in gynoestium and larger anther in plants with extrafloronectaries, and they might have a larger size. So here plants will have higher gynoestium than anthers, so no outcompatibility. So that's all the questions that I am trying to answer with this grant. I want to thank you all for watching this presentation. I want to thank especially SSE for providing this grant. And Kew Botanical Gardens, where I visit and where I stayed for two months collecting data. Here we have my contact, so if you have any feedback, suggestions, opportunities, you can send me an email or go to my website. And the take-home message from this presentation is basically that ants have a negative effect, especially on the flower visitation by bees. And we also found a negative evolutionary correlation between traits that mediate with pollination and protective mutualism by ants, mediated by extrafloronectaries. And also the ecological cost could be an important selective force. So we should consider the simultaneous effect of mutualisms on a focal individual instead of just evaluating one mutualistic interactions at a time. So thank you all for watching. That's it. I'm happy to answer any questions you might have.
Well, congratulations, Amanda. That's a really cool, and I think you're looking at many different things at the same time, which I think it's fantastic. You're taking into consideration several things. Um, I asked if anyone has a question about the your talk, and I have actually a couple of questions while everyone else um thinks about their questions. But so at the
It's
very
just
beginning,
should I stop sharing my screen or
uh I guess you can leave it like that. Maybe
okay,
someone's going to be asking something, you can go back and forth on the
okay,
slides.
nice.
Um, so at the beginning you mentioned that, so you're assuming that once the flowers have this very particular morphology, they are pollinated by bees because they're the only ones who can like activate this system, right? But I'm
Yes.
assuming that also in those forests or areas, ecosystems where these plants occur, you have like like different species of bees. And I was wondering if you mentioned at the beginning that you were also considering how much predation was occurring on the plants. So if you have, you know, a lot of predation on these plants, then you would expect them to have a stronger defense system. But I'm wondering if there's also, if you have a larger diversity of bees of potential pollinators, do you also get like maybe the costs for the plants are reduced? So you have, it's gonna it's gonna deter the pollinators, but also you have a larger amount of pollinators, not only individuals, but also different species. Have you have you overlapped or have you looked at diversity of bees in those areas where this negative relationship between the presence of ants and the extra floral rewards occur? Like,
Um,
is there any relationship between those two things?
I
Or you haven't
didn't.
checked?
Yes, I didn't, to be honest, because most of the data I'm presenting here is from studies that I collected data from some studies. So I didn't look at the effect of ants on different bee species. So the meta-analysis that I showed you, I can remember the amount of bee species, but probably some bumblebee species, honeybee species, they are all mostly studied. So I consider that ants could have an effect on all bees, and this effect could be the same. But indeed, maybe ants could have a larger effect on small bees than on larger bees. I'm not sure. And also, this was something that I was interested in the beginning of my PhD, but there was pandemic and I couldn't do any field work because of this. But this is a good hypothesis to test. I think hens have differential effect on depending on the bee. Also, for this system, the bus pollination system, uh, not all of bees can vibrate the anthers and get the pollen. So just some species can, some genera, I think. I think this is conserved in the genus level can vibrate the anthers and collect the pollen. For instance, honeybee cannot vibrate the anthers and collect the pollen, but bumblebees, stingless bees, they are able to vibrate the anthers and collect the pollen. So this vibrating species might be more affected than the non-vibrating species. One thing that is really cool about this system, this Chamaecrisis flowers, these flowers not all of them, but some Chamaecrisis species, they have a differential flower. They have a flower that has a C shape. But and once the bee vibrate the anthers, the pollen, some part of the pollen stays on this C-shaped flower, and this pollen are available to other floral visitors that cannot vibrate the anthers. They are just collecting the resource, but they might not stay on the flower for a longer time. So it's possible that ants might have a larger effect or a stronger effect on vibrating species than non-vibrating species, or also on smaller species than larger species.
So another question that I had, so when you mentioned that at the very beginning, you mentioned that the ants might reduce the amount of time that the bees spend on the flower.
Yes.
I'm assuming, because I have no idea about insects, but I'm assuming there are some insects that are faster in getting the resource from others. And I wonder if those are less discouraged by the ants to be pollinators. Is there any, I don't know if you know this, but if I wonder if there's any work on the amount of time the bees spend on the flowers, and that like those could be better pollinators for those species that have ants as protectors. I wonder if there's any information on that.
Um, Indeed, some pollinators they have they stay in the flower for a longer time. They pollinate they require more time to get a resource. So in Chemichrysalids stipulo, we conduct some experiments. And we show that the smaller species actually stay less in the flower. They have for a shorter time than the larger species. So I think ants could have a stronger effect on pollinators that stay longer. And this is also a really cool thing to evaluate. So you're giving me lots of ideas
Yes,
of
I'm just
things to evaluate in the
like your talk
future.
just started this kind of, my brain is going through what if we if you measure this, or maybe this, like I wonder how many other things could be interplaying here. It's really cool system. Anyone else has a question for Amanda?
Hi.
Hi.
Can I ask you a question? I'm Nonoko from Duke University. And can you go back to the slide showing the Q data describing many dots on the global map?
The slides. I didn't hear well which slide it is.
The slide which showing many dots on the global map.
No.
Yeah,
Yeah,
you just I think it's
just
I passed.
the map
Like the
with all the
distribution
purple
of the
dots.
specimens. Yeah.
That one.
yeah.
This?
Yeah,
Okay.
actually, I'm curious about the concentration of the dots because, yeah, it seems that the dots, most of the dots are concentrated on the southern hemisphere. So, why do you think that any kind of do you think that there is a possibility that we can get the data from the northern hemisphere?
Okay, so this clade is basically distributed in the neotropical areas, and I think that's why we have more data from the southern hemisphere. There is a few species that occurs in the northern hemisphere. I think in the US, for instance, there is only 2 or 3 species. We don't have any Chemichrysalids species in Europe. So that's why this clade it diversified in Neotropic, especially in America. So that's why we have more dots concentrated here, especially in Brazil, where we have lots of groups working with this genus. But we also have a high diversity of this Chemicrysta in Brazil. That's another reason why I chose this plant genus. I think more than 200 species occur in Brazil, 250.
Oh, thank you.
Anyone else with a question?
Yeah, I just wanted to ask, I think you mentioned this briefly. There's other systems where the ants are interacting with the plant that are not the extra floral nectar. And
Yes.
then, so there's other reasons why ants crawl on plants then. And then, are there any other systems that make the plant bee pollinated exclusively besides this particular system?
Okay, so protective mutualisms can be related to ants housing plants. Oh, sorry,
Plants housing
plants
it.
housing ants.
That would be cool
Yeah,
though.
yeah, that would be
Mhm.
really cool, but no, that not happen, I think.
No, I'm disappointed.
So some plants provide nests to ants like acacia, macaranga. So this is a really particular system and mostly obligatory system. So ant species and plant species, they are really constrained by the species. So there's one species that occur in this plant, there's one plant that occurs with this species. And the exophrynectaries, it's a more generalized opportunistic system because ant plants are secreting exophrynectaries all the time, all the time, all the time. Of course, not all the time, but when there's high power pressure, it's induced defense. So they secrete more exophrynectary when they are consumed by herbivores. But this is a generalized system. So one species of plant can be visited by multiple types of ants. And ants vary in a lot of traits, including aggressiveness. So some ants are really good protectors, but other ants, they are not. They see the herbivores and they're like, okay, you're here and I don't care very much about
Whatever.
you.
Mhm.
So they don't protect the plant, so they don't repel also the pollinators.
I see. Yeah, I was just wondering if the relationship existed between other ant and plant interactions, and then also on the other side of it, if there's other ways that plants become exclusively pollinated by bees besides this special stigma way. Like, are
Mhm.
these additional species that I know you've surveyed enough, you don't want to measure like everything out there, but like those are other analogous like species, right, for this phenomenon potentially. If those exist. I don't even know if they exist.
Yes. Um, but I couldn't um, think about any other trait that could be related to exclusive pollination by bees.
Oh, okay.
In this case of the type of resource that plants provide to ants, like housing or just food. In this case, I think that plants that how nesting ants and with if within their domations, they might have other type of ways to repel the ants from flowers. For instance, some plants have
Mhm.
volatiles that repel ants from flowers, though there is no conflict between ants and bees. Um, some plants provide oil, and I think that only bees are oil collected. But I couldn't think about a trait that I could measure and say, yeah, okay, this trait is exclusively related to bee pollination besides buzz pollination. But of course, there is more plants that are pollinated by bees, but they don't require just bees, maybe.
I see. I see. Well, thanks. The talk was really great, and I
Thank
like the
There's
you.
one more
water
question.
bottle as the prop. And we got one question, one more question
Yes,
here.
Mary Jane has her hand up. Go ahead.
But if you're out of time, it's not a very big question.
It's okay, Mary Jane. Go ahead.
Well, early, Amanda, early in your talk, you mentioned putting the reward. I don't remember whether it was extra floral nectary or the flower at the end of a pedestal. And I was interested in that because my specialty is studying social wasps, including some tiny wasps whose predators are ants, and that's true of most wasps in the neotropics. And a number of social wasps, Vestidae and Specidae, who build tiny nests, put their brood and the nest at at the end of a pedicel. It's quite a common pattern. So I was interested in that idea that you had of maybe having a pedicel that would be a barrier to ants or something that would keep them away from the reward. But then after that, you didn't mention the pedicel, despite your admirable interest in the morphology of this whole thing. So I wondered why you didn't answer it, didn't mention the pedicel again.
I couldn't hear very well. Some of the hosts could. I'm sorry, Mary Jane, I don't have my earphones.
Oh,
So the question
Uh,
was basically how you mentioned at the beginning the length of the pedestal as one of the strategies to get ants away from the flower, and then
yes.
you didn't follow up on what you found about this.
The size of the pit cell?
Yes, right, Mary Jane? Did I?
yeah, that's correct. Just the length of a pedestal that would make a long bridge for the ants to cross would keep the reward away from the ants, as it does in the nests of social wasps. That's why I was interested. So maybe you didn't see long pedicels in the plants you ended up studying. So that's why it's a very small question.
Amanda, did you get the question? So basically, it's in the genus that you studied. Was there a difference in the length of the pedestal? Was it a character?
Yes.
Was there any effect on that on your results?
Yes, there is. I think I just used one picture showing the thing I was measuring, and I didn't show pictures showing the variation. But yes, some plants have basically, I think maybe is a pet cell, but maybe we can see it. But yeah, I didn't. But here we have like a huge pit cell on this length of the pit cell, but these are smaller. I should have put more pictures here. I'm sorry. But yeah, there is a variation. I didn't run an analysis yet in my genus. These are all my predictions. For the angiosperm, I don't think I have a data set with the pet cell size of plants in general. So, but for Comicrysa, there is a variation, but I didn't run the analysis yet because I'm collecting the data yet.
Okay, thank you. If you ever do find a correlation, I will be interested in it. Thank
I
you.
will let you know, Mary Jane. Thank you so much.
Amanda, thank you so much for your talk, and thanks everyone for coming. I think it's 12:04 for me, but for you it's 1:04 already. So thank you so much everyone for coming, and Amanda, congratulations. Your work was really, really interesting, and it's definitely opens the door for many other questions regarding the system.
Thank you.
Okay, bye bye everyone.
Bye, guys. Thanks, Amanda. Great talk.